Welcome to the LemnaTec Academy. Depending on your experiments, the analysis results are self-sufficient or not. Often you have various cohorts of samples that differ in things like genotype, treatment, etc. In this video, I will show you how to add this kind of information to your database. If you combine the information that defines your cohorts with the measured data from your analyses, you could create graphs like this. We created these graphs on the Graphs and Plots page in LemnaExperiment. In LemnaExperiment, we call this information sample annotations, and they are stored in the database alongside the measurement results. But how do we get this information into your LemnaExperiment database? There are two places of LemnaExperiment involved. The Edit Experiments page. It defines which annotation columns will be available. On the Samples Annotation page, the actual values of the annotations are edited or inserted. There are two major ways to enter annotations. You prepare an Excel file and import them from it, or you enter them manually. You may add them before recording your data. In this case, you must use the Excel file import. Or after recording, but before you start your analysis, or after your analysis. Annotations are on a per snapshot base. This is usually overkill, but sometimes you have external conditions that are changing deliberately over the course of the experiment. With this approach, you can document this in the sample annotations as well. On the other hand, annotations are on a per sample base, not on a per vessel base. If your vessel contains only a single plant, then this makes no difference. However, if you're working with multi-well plates or plant trays, then a single vessel contains multiple samples, and each of them may have different annotations. For example, you may run an experiment with different fungicides in each row of a multi-well plate and with different concentrations in each column. There's one more thing to consider. If you run an analysis, this analysis creates results assuming a certain set of samples on the image, either just one or multiple samples. And this set of samples must match the sample annotations. If they don't match, this is reported as an inconsistency when you submit the analysis job. If there are no annotations defined for a snapshot that you want to submit for analysis, then LemnaExperiment automatically creates them. This is done the following way. If there are prior annotations for the same vessel in the same experiment, these annotations are copied. This way you need to annotate only the first recording run. That is, unless you have annotations that change over time. If there are no prior annotations, then annotations are automatically created using the default values you have defined in the experiment itself. Even if there are no annotation columns defined in the experiment, we will still create the structures to carry them, but without actual values. This is done for technical reasons. However, this comes with one danger. If you create an analysis job for a snapshot that is not yet annotated, this automatic will create these annotations for samples as defined in this analysis. But if you've selected the wrong analysis definition by accident, then this may create annotations for the wrong set of samples. If you later try to submit an analysis with the correct analysis definition for this snapshot, LemnaExperiment will not let you do it. Instead, it reports an error about inconsistent sample, as we have seen on the previous slide. This is of course only an issue if you have analyses for multiple samples on one vessel in the first place. But there's hope. LemnaExperiment has some options to correct this. However, I will explain them in another video. With all this background knowledge, we may now look into the software and how everything is handled. First, I like to handle the manual case. The first thing to do is to define the annotation columns on the experiments page. We open the editor for the respective experiment. Here you see the list of annotations I already have defined. We may add now another one. Each annotation column has obviously a name, a type, either text, integer, decimal, or Boolean, a default value, and optionally you may define a list of allowed values. These values are entered by typing them into the menu title and press return. They will then disappear and be an option in the menu. If you define values in this list, then only these values are allowed in the annotations for this column. Otherwise, all values allowed for the type of the column are allowed in the annotations. In the manual case, you can annotate only snapshots that have already been recorded. If they are already analyzed, then the system may have created them automatically, as I have described before. You may now modify them as needed. If you want to edit a value, you have to double-click its cell. Now you can enter values here. You see a completion list from which you may choose. Or you enter a completely new value. If we do this on a column with predefined values, these values are listed in the completion list. You are not allowed to enter other values. You can use the filters in this table to select a certain subset of samples. For example, we need to change the value of the concentration column, but only for samples in the 6th column of the microtiter plate. And only for plant type 2. Let's consider by accident we gave them the double dosage, so the concentration is 3.0. Now we can copy this to the other rows using the usual copy and paste shortcuts. Ctrl+C. We may use Control-click and Shift-click to select multiple entries. Now we can paste this value to these cells. Let's assume a slightly different scenario. Assume we gave each row of the MTP a slightly different concentration. A6 gets 2.2, B6 gets 3.2, C6 gets 2.8, and D6 gets 3.9. Instead of entering all this data for all the columns to follow, we can copy them at once. First, we select the first 4 rows, press Ctrl+C, and then we have copied them to the clipboard. Now we can select the other rows and paste the values to them. You see they are copied, preserving the pattern of the first rows. All these changes are immediately copied to the database. Therefore, we have no undo function, at least yet. If there are no annotations yet in the database for your experiments or parts of your experiment, then you need to create them first. This can only be done for snapshots that have already been recorded. The Arabidopsis experiment is such an experiment. We use the Create Annotation button to create annotation. This opens a new dialog. On the top, you can select the experiment to which you like to add annotations. With the second menu, you select which vessels to consider, either only those vessels which are not completely annotated. That means that all vessels are excluded which have at least one snapshot that has been annotated. This could make sense if you have annotated the first snapshot and use the aforementioned automatic to annotate the rest once you create an analysis job for them. The other option is to include those partially annotated vessels as well. On the left, you find the list of all vessels that needs to be annotated. You can select the vessels you want to annotate. On the right, you select the vessel layout to be used. In other words, which samples to be found on the vessel. If you have only single plants in your system, then the first entry is probably the only one. If you press Now Create Annotations, the software will create annotations for all snapshots of the selected vessels that are not annotated yet. The values used for these annotations follow the same logic as described for the automatically created annotations. As you can see, we have now annotations for the Arabidopsis experiment. As I said before, you can also import annotations from Excel files. You can import these data before recording. In this case, the snapshot will be registered to be used with the next data imported for the respective vessel. You can import these data after recording. Then you must supply the correct snapshot name to identify the snapshot and its samples. Or you can import them after the analysis. In this case, this will change or update annotations in the database and/or add new annotation columns. I will demonstrate this with an example for case 1, importing annotations before recording. On the sample annotations page, we press the import button. Now we select the Excel file. Now the import process starts with an analysis of your Excel file and check if its content is consistent with the database. Once it has finished this, we get a report. In this case, we are told that we have one new experiment, but no changed experiment, and 2,400 new annotations, but no changed or updated annotations, and that there was no error, warning, or info messages. If we have errors, this might look like this. Now we can press the Proceed button and import the annotations. This is a fairly small set. Really large sets may take longer. For details on the file format, you can check the online help. Under Technical Details, you find the section Registration File Formats. You can also export an existing annotation table using the Export button on the sample annotation page. You may use this as a template. In this video, we learned how to add sample annotations to your data in LemnaExperiment to cover additional external information on your samples and cohorts. See you in another software training video.